two-dimensional electrophoresis. It may be called as 2D gel electrophoresis. Now, suppose we want to study the structure of proteins in order to evaluate their function, how we can study. So, like the nucleic acid, we have to isolate the protein molecule and then purification step can be performed. So, in this case, the proteins can be extracted from the cells as well as from any tissue. And after extraction, it will represent the crude extract that contain variety of proteins. कि इसमें बहुत सारी प्रोटीन्स होंगी. हमें नहीं पता कि जो हमने प्रोटीन टारगेट की है, जो एक्सप्रेस हुई है, वो कहाँ पे है, आल्दो वो इस मिक्सचर के अंदर ही है. तो उसको हम अब कैसे सेपरेट कर सकते हैं और उसका स्ट्रक्चर स्टडी कर सकते हैं. सो like the RNA molecules that can be separated by running on the gel. So same principle it can be applied for the proteins, that the crude extract of proteins, it can be separated on the gel so that we can get different bands, and then those bands can be excised and can be further evaluated for the structure by applying other techniques. So as far as this 2D gel electrophoresis is concerned, it can be combined with SDS page, like sodium dodecyl polyacrylamide gel electrophoresis. And what is the principle that we can use for the separation of proteins? So as the name of this technique is indicated, that here the two-dimensional approach it is being used, that protein first run in one direction and then on other direction, so that proteins molecule, they will be resolved or separated from one another. So in this case, as far as first dimension of the movement of the protein is concerned, so after loading, the proteins, they are being run in the gel that is having a pH gradient, like suppose pH gradient that may be 3 to 12, or some narrow ranges there like 5 to 6. So that on the basis of their electric charge, protein molecules, they will be separated in the pH gradient. And this technique or principle, it may be called as the isoelectric focusing. Now in isoelectric focusing, proteins will move in the gradient of pH until they are in their isoelectric point. That Pi value it can be indicated. Isoelectric point. Isoelectric point is that situation where the net charge at a given pH or surrounding pH, it will become 0. So that proteins, they are resolved on the basis of their isoelectric point in the gradient of the pH. So proteins, they have moved in one direction, that may be towards the cathode from the anode, depending what charge they have, until they reached isoelectric point. Now proteins, they are separated in one direction, and then a perpendicular charge is applied that will result in the isolation of proteins on the basis of their molecular weight. Now what is the actual principle behind the 2D gel electrophoresis? As in this diagram, it has been indicated that we can run the protein in pH gradient. Suppose we have loaded our sample here. At this corner the pH is high, here the pH is low, so we have established a pH gradient. Now molecule will move in this direction. So it is actually a gel tube where we can going to get the separation of the proteins in the pH gradient, so that proteins, they will be resolved, they will be separated. Their circle size indicate their molecular mass. So proteins have been isolated, and then perpendicular charge is applied at right angle. So first, according to the first direction. So the charge no another direction, so that no proteins they will be resolved in another direction, so that they are well separated. So this is the basic principle. So actually what is happened? So this is the protein separation from mouse liver, and this is on a wide range of pH that may be 3 to 12, and this is narrow pH range like 5 to 6. Narrow pH range result in greater resolution of the spots. So spots, they are well separated as compared to wide range of pH that spots are close together. So once spots they are separated, they can be further excised for analysis. So this gel, it is stained with nitrate, silver nitrate. So by using 2D gel electrophoresis, thousands of proteins they can be studied on a single gel, like 2000 to 3000, and sometimes it is reported more, around 10,000 molecules they can be studied. Although the most abundant classes of proteins can be studied by using 2D gel electrophoresis. One limitation is that कि जो rare proteins हैं, जो कि comparatively copy numbers में कम है, उनकी detection जो है 2D gel electrophoresis से वो उनकी resolution नहीं होती, उनकी detection इतनी अच्छी नहीं हो पाती. तो बात सिचुएशन में ये जो 2D gel electrophoresis, it is very important कि जो differentially regulated genes हैं, कि जो express करती हैं under particular set of condition, उनकी डिटेक्शन में ये बड़ी वैल्यूएबल है. लाइक डिजीज सिचुएशन में कि अंडर डिजीज सिचुएशन कौन सी प्रोटीन एक्सप्रेस हो रही हैं और अंडर नॉर्मल कंडीशंस वो एक्सप्रेस नहीं होती. सो 2D gel electrophoresis, it is very valuable technique for the resolution and separation of thousands of proteins on a single gel.